analyzing heme binding efficacy and alpha-helical propensity. So I wanted to give a brief introduction on peptides and heme. So peptides are constructed of amino acid building blocks. Here are 20 of the most or 20 common amino acids, and essentially based on which amino acids you use in your peptide or the sequence of them, it'll have effects on the structure that the peptide ends up assembling into, and also the different properties of the peptide. So three of the most common secondary structures of peptides are alpha helix, beta sheet, and random coil. So alpha helices are like this graph here, which is like a coil. Beta sheets are the other one where they kind of stack on top of each other and form these sheets of peptides. And then random coil is just when they aggregate together and form these clumps of peptide. So this is heme right here. It's kind of this ring-shaped molecule with an iron in the middle. And heme has the ability to bind to some proteins, and these heme-bound proteins are fundamental in many biological processes. For example, hemoglobin is actually a heme-binding protein, and it's used to carry oxygen in your blood. This is a visualization of a protein that is bound to heme. So you can kind of see like the coil, so it's an alpha helix. This cyan bit here is histone, which is an amino acid that binds to heme. And then this is the heme molecule with the gray iron in the middle. Okay, so some motivation on why we wanted to study these materials. Basically, something that I thought was really interesting is that since heme has iron in the middle, if these proteins kind of come together and form these long filament-like structures, it provides the opportunities to have a biological material that can conduct electricity. So wise, it also has antimicrobial possibilities. And our goal is basically to find certain patterns in the amino acid sequences that would create peptides that self-assemble into alpha helices and that would actually bind heme. So onto the methodology. The first thing that we had to do was synthesize these peptides that we could do experiments on. So we used solid phase peptide synthesis, and we synthesized 16 amino acid long peptide chains. We had to rationally design certain sequences that we wanted to actually synthesize, and we did this based on, we considered charges, hydrophobicity, and then obviously including a histidine so that it could actually bind to heme really successfully. Here are some examples of the peptides that we designed. So in the first three, you can see the K is lysine, the L's are leucine, and the H is a histidine. So we're essentially increasing the amount of lysine at the beginning of our peptide, and lysine is actually positively charged. So kind of what we're doing is we're creating a charged head and then a hydrophobic tail, and then histidine in the middle there. Okay, so we used ultraviolet displacement spectroscopy to help us determine how much heme is bound to our peptides versus not bound. And essentially, we just shined certain wavelengths of light in the ultraviolet invisible spectrum through our sample and then into a detector, and it would give us, based on the absorbance, it would give us these spectra. This is an example of a heme-bound spectra and then a non-heme-bound spectra. So you can kind of see a problem arising, which is that they look decently similar, and so it's kind of hard to qualitatively determine. For example, if 50% bound, it would be a combination of these two, and it would be hard to actually see how much really did. So we used linear combinations fitting process to kind of get more of a quantitative number on that. So here's an example of that being used. You can see the blue is the spectroscopy, the spectra that we actually got. Red is the fitted line, and so we found that this gave about 45% heme binding. And since the lines are pretty close together, we know that our fitting is decently accurate. Okay, and then so to determine secondary structure, we used circular dichroism spectroscopy, where it's very similar to UV-Vis, except instead of wavelengths between ultraviolet and visible light. We polarize light circularly and then shine that through. And depending on the absorbance again, we get spectra which correlate to different structures or things like that. And we also use linear combinations again to kind of get numbers on how much percent is actually structure. This one you can see is about 2.9% helical, 33% beta sheet, 27% random coil, and 37% aggregated helix. And again, the lines are pretty similar, so we know we got a decently accurate. Okay, so there's a lot of, if you imagine 16 different places where you can put amino acids, 20 different options, there's a lot of possibilities. There's a lot of different things that affect whether it'll bind heme or not and what kind of structure it'll have, and we can't rationally determine all of those. So all of the data we collect will be put into a machine learning model that'll help us figure out these patterns. But along the way, some of the ones that we realized, well, I'll present two of them. One of them is, so these are UV vis spectra, so basically telling you whether it bound heme or not of some of our peptides. This one is lysine, so there's 2 lysine on the head. This one, there's 3 lysine on the head, and that one there's 4. So if you imagine like the blue ball with the blue stick I showed before, basically the blue ball is getting bigger and bigger, and the charge on the head is increasing. And going along with that, we also see that the heme binding is increasing. So like a bigger charge stick correlates with more heme binding, and we think that this is because so our peptides, the green part is hydrophobic and the blue part is charged, and so basically they're going to form these structures where the hydrophobic part wants to get away from the water. And once these structures form, it's going to be really hard for heme to kind of get into there and bind with histidine in the middle here. So what we speculate is that with a larger charge on these blue parts, they're going to be repelling each other more, creating a more like dynamic and flowy structure, allowing heme to actually get in there and bind the histidine more. Another pattern we noticed was that, so this is also increasing the charge on the head. This time it's a negative charge, but then the alpha helice percentage actually decreases. And we think that this is also similar to how a larger head will make it more dynamic. We think that it'll get too dynamic to where our structures start falling apart. So this is a cryo-EM image of the peptide sequence that has 2 glutamic acids on the head, so it's a relatively smaller charge on the head. And you can see that the structures it forms are very like distinct and like they are circles. But then over here, once we get to 4 glutamic acids on the head, it'll form these balls, these spheres, but also a bunch of filaments, so not only alpha helices, but also beta sheets. And then future research, so basically my part of the project is just collecting a database of data, I guess, on peptides. And what we're going to do with all this data is put it into a machine learning model that could help us suggest more sequences to do more experiments on and collect more data. And then, so it's kind of like a cycle of research. Nathan, who's presenting after